Dzień dobry Państwu.
Dzień dobry.
Dzień dobry.
Poczekajmy jeszcze chwilę, może się jeszcze parę osób zaloguje.
Dzień dobry.
Dobrze, a w międzyczasie spróbuję uruchomić prezentację. No i zamarzłem. Mnie kompletnie odcięło. Czy słyszycie mnie teraz?
Tak, słyszymy?
Tak.
Ja zamarzłem na tym całym ekranie, bo coś się tu dzieje. O dobrze, może się teraz uda. Próbuję uruchomić. Dobrze, tak to zamknę. Czy teraz widzicie prezentację? Małe slajdy, czy nie?
Tak, widzimy.
Tak, widać mamy slajdy.
To jesteśmy na dobrej drodze. To już jest jakaś szansa. Dobrze, teraz spróbujmy zrobić pełen ekran. Coś się dzieje, ale nie do końca. Dobrze, jestem na slajdzie 6. OK. Dobrze. OK, jako tako wszystko działa. Zobaczymy, jak dalej pójdzie. Także zaczynamy wykład trzeci. Może na wstępie, it's supposed to be in English, na wstępie może zrobimy małą po polsku też podsumowanie i po angielsku. So, previously we just talked about the replicon model. You remember the structural gene for replication initiator, the protein which has to bind to origin of replication, and we discuss different methods for the analysis and identification of replication origins, clonal analysis, 2D gel analysis, features. Please remember about the features of replication initiators, AT richness, DNA unwinding elements, compositions containing binding sites, multiple binding sites for replication initiators as DNA box sequences, and also the feature which is unequal distributions of A's and T's and G's and C's, especially AT's for the among the top and bottom strand, and bioinformatics analysis. We also discussed the structure of replication initiator for as an example of DNA protein, and this is what we just discussed, that this is just the two domains. One domain is a DNA binding domain, and the second is AAA+, so ATPase, various ATPase activities of DNA protein with ATP, which is important for interaction with single-stranded DNA, and also we discussed the models showing the filament of DNA with open and closed structure, important for the unwinding of the replication origin with a specific this DNA unwinding element site, which is AT reach. Okay, so this is just the important features. Also, I would like to stress your attention on methods. So we discussed DNase 1 footprinting as a method for identification of proteins interactions with DNA, and identification of actual sites of these interactions, sites on the DNA molecule. Then we discuss P1 footprinting, which is a different type of footprinting based on the nucleolytic activity of P1 enzyme, and this is combining with a primer extension reaction gives you the possibility to find the single-strandedness of and identify the sites when the DNA is where the DNA is single-stranded. Okay, we also mentioned about the plasmon resonance as a real-time analysis of intermolecular interactions, and the intermolecular interactions between DNA and proteins, or between proteins and proteins. So whatever you would attach on the sensor in here, then you can identify the interaction. So it would be association and then dissociation, regeneration of the system, and then you can just do next interaction analysis. And this is, of course, in the time, and the response units are giving you information about the mass on the sensor chip, and the mass relates to the number of molecules which is attached. So knowing the number of molecules which are attached on the sensor at the beginning, you can identify the number and kinetics of you can firstly identify the mass, increase of the mass, and then the number of molecules interacting analyte. Okay, and then we discuss about different sites for interaction with DNA protein, and you remember that those sites were ATP and ADP dependent, and DNA protein in two different forms can bind different sites, and also at the end of the lecture, I pointed out your attention on DNA trios, which are specific sequences important for the opening reaction. Okay, so let's move to today's lecture, and at the beginning a few words about the regulation of chromosomal and DNA replication in bacteria. And as the replicon model says, again, the same situation. So the gene, the protein, and the interaction. So the system looks simple, but it turns that it's not that easy how the replication initiation, it was not easy, and it's still not easy to understand, to fully understand the mechanism for the regulation of DNA replication initiation. Okay, so we have very many systems, and I will try to go with one by one and showing you the complexity of this regulation. So first, we mentioned about the DNA-A, that DNA-A exists in two forms, ADP and ATP, and those two forms differently interact different binding sites. It's very important, but also it was shown that without DNA-A interacting at OREC within this ATP binding sites, the replication initiation is impossible. So the amount or the concentration of DNA-A in ATP form in cell is critical for the replication initiation. And there is a specific system which involves two proteins, beta clamp. We will talk about the beta clamp later on. It's a part of replication complex of a polymerase. It's a polymerase subunit. And the other protein is HDA. It's HADA, and the HADA interaction with beta clamp and interaction with DNA-A at the replication origin and during DNA synthesis converts the DNA-A from ATP to ADP form. Then acidic phospholipids bound DNA-A ADP form and can convert ADP to apoenzyme formation. And then this apoenzyme can be converted again to ATP form. So this is just a RIDA, regulatory inactivation of DNA-A. And this is turned on after replication initiation, which is supposed to prevent reinitiation, quick reinitiation, because it converts the replicationally active ATP form to replicationally inactive ADP form in the system. Pretty complex, but this is not enough for the complexity in cells, and we'll go with the next systems. So I mentioned RIDA. We are done with RIDA, and now let's look at the other one, which is DAT-dependent DNA-A ATP hydrolysis. So this is the other system which converts DNA-A in ATP form to DNA-A in ADP form. Okay, so this is this conversion, this type of conversions, and this is just a RIDA and DDAH. DAT dependent and DAT-A are specific binding sites which are found in bacterial chromosomes. To those sites, DNA-A in ATP form binds and is converted to ADP form. Okay, so this is this, and the other system is DARS. Okay, and the DARS is DNA-A reactivation or a nucleotide exchange. So it works a little bit opposite comparing to DDAH and RIDA system. It converts DNA A in ADP form to DNA A ATP form. So it's a reactivation reaction which also have an influence on the replication initiation. And this is how it looks like. You see that the structure of DARS was specified. These are specific sites which accommodate many DNA molecules, which are posted in here. And this is maybe better. You see those DNA box blocks, which forms the DARS core. And you see the ADP DNA interaction with those sites. This is ADP. And after these interactions and in the presence of DIAA protein, at least 3 molecules of DNA interact together, but the interaction among 2 are important, which release ADP from the complex. So actually it creates the APO enzyme at the beginning, and then this APO enzyme is loaded with ATP. Yes, so this is this reaction. Okay. Okay, so ADP is released and ATP is loaded. This is just the system which works. So actually it's a circle. It's a circle which converts ATP form of DNA to ADP form to DNA, and then ADP form to ATP form, and again, and again. And these fluctuations of ATP, ADP form of the DNA replication initiator results with inhibition or activation of DNA replication in bacteria. Okay, so, but on the top of this is something which is very important. It's proteolysis. And we found that long protease has an affinity of DNA A protein in ADP form and can degrade it in the cells, but only in the presence of poly-P. Polyphosphates are synthesized in cells during stress. So you can imagine that if stress is applied on bacterial cells, then this poly-P is synthesized. The poly-P forms specific granules, and then DNA A and long interacts with this poly-P scaffold, and DNA A in ADP form is degraded. I will show you a few experiments. So this is just the degradation of DNA A after applying the stringent response. This is just the amino acid starvation, stationary phase. You see the depletion of DNA A protein, and also the experiment showing interaction of DNA A protein with polyphosphates and interaction of long with polyphosphates. So you see that this 2 proteins form these complexes, and this is just important for the degradation of DNA A. But why did this so important that the long protease degrades ADP form? I told you that the ADP is replicationary non-active. Well, it's during the replication arrest during stress conditions, and that was in E. coli. This RIDA system works and converts DNA A from ATP to ADP form, and this prevents reinitiation of replication. However, the round of replications which were already activated has to be finished. So it's like converting ATP to ADP and then degrade it to prevent reactivation. Okay, so that makes sense, especially that I will tell you just within a minute that ATP DNA also has another important feature. It inhibits its own transcription. So it makes sense because then the concentration of DNA in starved cells under stress conditions is lowered, and DNA ATP still can inhibit its own transcription of its own gene, and these new DNA molecules are not synthesized during stress. Okay, so in other bacterium like Caulobacter crescentus, this is not polyphosphate, but it was demonstrated that lone Caulobacter crescentus lone degrades both ATP, ADP, and ATP DNA protein, but unfolded substrates are stimulated this activity. So unfolded substrates are also present in bacteria cells during stress. So this is another stress alarm which actually triggers protease activity towards degradation of replication initiator. Okay, so this is just the ATP, ADP, and we can say that also protease can work in here and degrade ADP DNA form, but ATP can also is important for the, as I mentioned, DNA gene autoregulation. So it prevents transcription of DNA protein. It's inhibited by titration to that sites, but also the other protein prevents, which is called SEC-A, it prevents interaction of DNA in ATP form with, and now I would like to pay your attention, with newly synthesized DNA. So it's SEC-A dependent mechanism for inhibition of DNA replication, and SEC-A is a protein which interacts hemimethylated DNA, hemimethylated by DAM methylase, within the GATC sites. And how it works, so when the new DNA is synthesized, one strand is methylated, but the newly synthesized strand is non-methylated, and it takes time. So it's just a very transient and dynamic situation that we have hemimethylation just close to the replication origin, and SEC-A protein binds this hemimethylated DNA, newly synthesized hemimethylated DNA. So that would be, this part would be fully methylated, but this part, after progression of replication fork, is going to be hemimethylated because only half of the DNA is methylated, and the new strands are not methylated yet. So then the SEC-A binds there, and that prevents the interaction with ATP DNA, and this prevents reinitiation, immediate reinitiation of replication. Okay. Oh gosh, it's a little bit hard, but I think you managed. So what we have? ATP, ADP conversions, DNA degradation, and it could be both ATP and ADP form, as in Caulobacter crescentus, but in E. coli only ADP DNA is degraded by lung. Okay. Then we have the other system, which is based on hemimethylated DNA and SEC-A protein, which interacts hemimethylated DNA, which prevents ATP DNA to interact replication origin. And also, the ADP has regulatory autoregulation function to prevent transcription from its own gene. And I think this is just the DNA ATP protein transcription effects, and then ATP, strong repression of ATP, and weak repression of ADP, form of which binds to DNA boxes within the promoter of DNA gene. And you remember when we discussed the identification of replication origins, one feature was just the presence of a gene for DNA protein is close to the replication origin, which altogether makes sense considering the regulatory mechanisms. I think this is just the list about all these factors, which dat locus, DNA titration, Sec-A, RIDA, and DAR sites, DDAH, and PDEP is poly-P-dependent DNA degradation during starvation in E. coli, and DNA degradation by long prototoxic stress in Caulobacter crescentus. Okay, so what about the DNA and extrachromosomal genetic elements in bacteria as plasmids? So we already discussed that in chromosomal DNA, those DNA boxes are going to be present in different sites, but the concentration of them are close to the replication origins. But when you look at the extrachromosomal genetics, so you can identify DNA unwinding elements, iterons, we will talk about this later on, and DNA boxes as well. And you can see clusters of or single DNA boxes present in the extrachromosomal genetic element origins. Okay, so this is just a comparison. You see that the structural elements of replication origins are similar. So AT rich with DNA unwinding element, and then boxes all over the places in the chromosomal DNA and those clusters which are already mentioned, or in the extrachromosomal genetic elements. This is just the same picture, just pointing the DNA box presence in the replication origins. So then we will discuss what is the reason and how the DNA protein interacts those clusters. So if we have the cluster with replication origin, and this is just the wild type, so you see that there's like two pairs of DNA box sequences which are inverted, okay? So this is just one pair which is directed, the other pair which is directed, but those are inverted comparing to those. If you switch this number or make a deletion or flip the box position, that results, so this is just the wild type origin and the interaction of DNA protein. This is just gel retardation or electromobility shift assay, gel retardation assay. This is just the equivalent name for this. And you see different complexes formed by the DNA protein with a wild type origin. However, if anything is altered, then you see no interaction at all, or some different interactions, but then the bands are very faint and you see that something is going on, that the specific position is required. And now we will make an interpretation of those electromobility shift assay. So please remember how the electromobility shift assay works. You are taking DNA fragment, for example this, and then you make experiments with different reactions. So it's going to be one tube, the second tube, the third tube, and so it's 1, 2, 3, 4, and 5. And this is reaction number 2. Okay, number 1, 2, 3, 4, etc. And you see in reaction number 1, you have a piece of DNA and nothing else. Then you have a piece of DNA and particular concentration of DNA protein. And then with another tube and another tube, you have an elevated concentration of DNA protein. So what we see different complexes. What those complexes mean? They mean that in the higher concentration, you can see 1, 2, 3, 4 different complexes. So it is very likely that 1, 2, 3, and 4 DNA molecules occupy the site. Okay, and that makes the shift, electromobility shift of DNA in the gel. And now we have ORIT-A4. So you see that this is just the number 4 is deleted. There is no number 4 DNA boxes, and we don't see any retardation. So what that means? Well, it means that the A4 box is the most critical for the interactions with other boxes, and there is no interaction present when the box number 4 is deleted. Okay? And then this is just what you see. And now I would like to show you the next slide, and this is just another experiment when the box number 4 was just flipped. So we changed the orientation of the box, and this is so we have a wild type and the mutant DNA fragment analyzed, and you see a little bit different picture. So what would be your interpretation of this picture? So the first question is, does DNA protein interacts ORIV4 flip fragment, DNA fragment? Who knows? Does it interact or not? No, come on, are you there?
Looks like it does.
Yes, it looks like it does, because we see shifts. But this shift is a little bit different. So why is that? What do you think about this? I mean, just if you look at this bands in here and those bands in here, it's different, yes, obviously. What is the difference? Well, we can see that in the bottom panel, the band number 1 is the most intense, and then is weaker, weaker, and weaker. But in the top panel, the band number 1 is more intense. Band number 3 is even more intense comparing to band number 1. What do you think? Why is that? What would be your interpretation? Well, so this is, I am showing this to you because it tells you that it tells you something about the nature of interaction of DNA protein, and whatever protein you would analyze, this type of picture indicates that the interaction is cooperative. Cooperative. Yes? And what that means that the interaction is cooperative, it means that unless one DNA molecule, one protein molecule is bound, then it's much easier to interact the second molecule, okay? And then the third molecule, because those proteins interact, those protomers interact most likely with each other. So it's a specific cooperative binding. And if we have the same situation, but there is only one interaction, one protomer of a protein interacting, it goes on and off rapidly, on and off rapidly, on and off rapidly. But then when suddenly the other partner interacts, the complex is more stable, and this is the reason that this band is better visible comparing to uncooperative interaction. Okay, so this type of picture tells you about the cooperativeness of the interaction of proteins within the DNA molecules. Okay, so what are iterons? And iterons are a binding site for the plasmid-encoded replication initiators, and you see based on this picture that there is a multiple binding site, not one, not two, but very many. There is only one example with 3 binding sites, but you may have 5, sometimes 6, 4 binding sites, and usually in contrast to DNA epoxies, those sites are directed repeats, so they point one direction. So it's going to be like a sequence directed after sequence, the same repetitions. This is just the typical for extrachromosomal genetic elements, that those are binding sites which are directed. And this is just to show you that REP, plasmid replication initiators, REP proteins interacts, and this you can assume that this is going to be D and is 1. Okay, I should DNA is 1 footprinting, showing the interaction sites, identifying interaction sites of replication initiators within the directed repeats 1, 2, 3, 4, 5 in here. And then, okay, I'm sorry. And then this is just gel retardation or electromobility shift assay showing the interaction of replication initiator with directed repeats. Okay, but when you do specific experiment which would which consist with 2 different steps, and the first step is size exclusion chromatography. Okay, so you are taking a sample with a replication initiator, and you do size exclusion chromatography. So actually you just select the fractions, okay, fractions based on their site of a protein inside, and then you take those fractions and you do gel retardation. So you add the DNA and you test the activity for binding. So you see that the best interaction with DNA is going to be, I don't know, 20, yes, those 3 fractions. Do you agree? 29, 30, 31. But if we would look at the size exclusion chromatography, so this is the fractions which are here. So surprisingly, less protein, better interaction with DNA. More protein, fraction 27, worst interaction with DNA. And these experiments raise the possibility that maybe replication initiator forms different forms, and it could be monomeric in fraction 30, 29 kilodaltons, but dimeric in here, and that dimeric form of replication initiator does not interact iterons, but the monomeric form does. And this is very true. If you compare the dimer interaction of rep dimer interaction with iteron containing DNA, it's pretty weak. And when you look at the monomer interaction with DNA, you see that the bands appears, and there is no question that the protein interacts with DNA fragment containing iterons. And we can even count number of iterons: 1, 2, 3, 4, at least 4, maybe even the 5th one in here. Okay, so the important information is that this is not that whatever replication initiator in whatever form interacts iterons, but it must be in monomeric form. And this is just experiment showing you that this monomer form could be purified due to the specific mutations, or you can convert the wild type dimer to the monomeric form by the introduction and incubation with molecular chaperones. And this is just molecular chaperone, which is called CLIP-X, E. coli molecular chaperone, and the conversion of a dimeric form of a replication initiator to the monomeric form of replication initiator results by the chaperone protein CLIP-X results with better interaction with heterons, and this is just the conversion size exclusion chromatography, and you can see that the dimeric form of a protein was converted to the monomeric form of the protein, and this monomeric form of the protein is much better interacts with DNA comparing to the dimers, to heterons. So this is very true in very different systems, that the monomers of replication initiator, monomers of DNA protein, but not the aggregated form, has to be converted by chaperones. This is DNA K protein, HSP70 member of a molecular chaperone, DNA K in here, HSP70 in yeast system, activating origin recognition complex, which is a eukaryotic protein for the firstly monomerizing it, and secondly this allows the associated of rep or DNA A or ORC proteins monomeric forms with replication origins. Okay, so this is something bizarre that in the prokaryotic, the replicon model says that there is again the gene, the protein, and the replication origin. However, I'm telling you that this is just the plasmid and the chromosomal DNA. And the chromosomal DNA has a gene for DNA A. And plasmid encodes gene for rep. And this rep has to bind to the replication origin. And DNA A has to bind to the replication origin to initiate the replication. So what is the role of these two factors, two replication, two binding sites, and two replication initiators during replication initiation? And this is the experiment actually demonstrating it. It's a little different. The third type of footprinting reaction, which is called potassium permanganate KMNO4 footprinting reaction. And this is based on the oxidation of potassium permanganate by potassium permanganate oxidation of thymines, but only in the single-stranded form. So then again, if there is a modification, oxidation, then you can perform primer extension, and this oxidation generates stops of extension. So then, because of labeling, you can just read the length of stopped DNA fragments, and the length of the stopped DNA fragments actually indicates the single-strandedness and the opening reaction. And you can see that this are those fragments which have been modified by potassium permanganate, and then the extension was stopped at those positions. And what we have different compositions of reaction with potassium permanganate, and we have the presence of rep and presence of DNA A. So when you look at the reaction with, I should look at this. Okay, DNA A only, so it's this one, this band, you don't see any stops. So it looks like DNA A protein at the plasmid replication origin is not doing that much. Okay, then when we look at the reaction with the replication initiator encoded by the plasmid, you see some modifications. So there is an opening, the destabilization within the dua. Yes. However, this destabilization is not that stable, or maybe not to the same extent comparing to the reaction where both Rep protein and DNA A protein were present. So this is just this one. Okay. So this tells you that both Rep and DNA A are required for the opening reaction at DNA unwinding site, and because of the sequencing and identification of this part, which was modified by potassium permanganate, it was possible to identify the sequence, and it was determined that this DNA unwinding AT-rich part of replication origin contains repeated 13mers. Okay, you see that there is L, M, R, and M2, and this is just specific consensus. You see that this consensus contains very many timings, and this is very similar with chromosomal and extra-chromosomal genetic elements. So all of them contain repeats, and all of them have some sequence similarities of those repeats, and there's like a very rich of timings on one strand, and this is a region which is highly unstable, and it's destabilized by the presence of DNA A protein in chromosomal DNA replication origins and DNA A and Rep proteins at the exochromosomal genetic elements. Okay, and this is just for you just to be able to compare different footprinting methodology and for what those methods could be used. So P1 and potassium permanganate for identification of single-strand parts of chromosomes, and DNase 1 footprinting for identification of proteins interactions with DNA. Okay, let's make 10 minutes break and then we will continue, okay? And okay, I have just to collect your presence, so please don't disconnect. I will try to do so. Okay, there's all of you are present here, so I will try. Okay, we are done. So 10 minutes break, okay? Or let's meet 1:15. Is it fine?
Yes.
Okay, 1:15. Okay, I'm back, so we can continue. Where we are. Here, no, sorry. I just lost my presentation on the screen. Where it is. Oh, I think. Can you see the presentation?
Yes.
Yes.
And the drawing as well?
Yes.
Okay. The problem is that I have 3 screens and sometimes I just miss. I should just write on the marker what is the screen number and then it will be easier. Okay. So just part 2 of the lecture. And so we will talk a little bit about the replication initiator structures. And we already mentioned a little bit. You remember DNA binding domain and a plus domain as parts of DNA A protein. And I would like just to tell you a little bit, just give you a little bit terms. So there is something which is called SNAPs, specialized nucleoprotein complexes, and these are complexes formed on DNA. And also the other term, OBPs, origin binding proteins. So these are the proteins which interact with DNA, and obviously all replication initiators are origin binding proteins, and the bacteria encoded DNA A proteins contains ATPase associated with various cellular activities domain, AAA plus, and DNA binding domain. When you look at the replication plasmid encoded Rep proteins, and then when you compare the monomeric sequence of monomers of Rep, and they consist with N and C terminal domains, and if this is N terminal domain, and this is C terminal domain, and when you look at this and you do the alignment, and this is just the secondary structure alignment over there and over there, so you see that N terminal domain is kind of like a little bit like a mirror of C terminal domain. We have B1, B2, alpha 1, alpha 1 prime, alpha 2 prime, B2, B2 prime, alpha 3 prime, and so, and it's helix turn helix domain and beta turn beta domain. So these are very similar when you look at the structure, tertiary structure. Then this is just the C terminal and N terminal domain of Rep E protein, and you see that there are some similarities. So this is just the alphas, alpha 4, alpha 4, alpha 3, and then the specific arrangement of beta 2, beta 3, beta 4, beta 2, beta 3, beta 4. So very similar, kind of symmetric. And this symmetricity is important for the interaction. The first ever published structure of replication initiator was Rep-E of plasmid encoded replication initiator. I think also replication initiator as ever is Rep-E protein, and the Rep-E monomer consists of the similar domains, and those similar domains kind of sits on the DNA growths. And this is just the N-terminal domain and C-terminal domain, and these are critical interactions. Okay, you see this. And this is top view. Okay, and you see that this is N-terminal and C-terminal domain. So this is just a single polypeptide with two domains, not two proteins, but there is symmetric structure, and this symmetricity is important for actually very many replication initiators. And this is superposition with CDC6, and this is position of superposition with Rep-E, and you see that the structure is kind of similar. And okay, also I should tell you that those domains contain specific motifs which are called winged helix domains, and this is just this WH abbreviation indicating those domains present both with Saccharomyces cerevisiae and Pyrobaculum aerophilus CDC6, so actually ORC proteins and plasmid proteins Rep-A, and also archaea proteins can contain those wing helix domains typical for the replication initiators. This is just also superposition of Rep-A and CDC6, and later on you could be able just to do stereoscopic view when you look at this and you concentrate your eyes to bring those two pictures together, and then you get the stereoscopic view of the structures of Rep-A protein and superpose with CDC6. And this is just another examples, Rep-E and CDC6 and DNA-A and CDC6, but the DNA-A is this triple A plus domains which are superposed. So in this slide, I am showing you that if you compare CDC6, this wing helix domain, the specific domain which is also typical for plasmids, and this is superposition of eukaryotic and plasmid replication initiator, then you see similarities in terms of a structure. But when you compare CDC6 triple A plus domain with DNA-A triple plus domain, you also see similarities. And this is just what's going on. So we have DNA-A protein, which contains AAA plus and DBD. Then we have plasmids, which contains wing helix domains. Okay, the one and the second one, and then combine the specific structure which sits on the DNA. But when you look at the eukaryotic replication initiators, ORC or from archaea, then you see that the similarity between the bacteria, and this is AAA plus part, and plasmid, and this is wing helix part. So it looks like eukaryotic replication initiators are combined with two different structures. One typical for bacteria, and the second one typical for the plasmid encoded replication initiators. So when you do phylogenetic relationship between origin binding proteins containing AAA plus domains, this is DNA A, and you see that there is some relation with ORC4, ORC5, CDC6, ORC1, and ORC1. So bacteria and eukaryotic replication proteins. But when you do the same relationship phylogenetic analysis based on wing helix domains, then the eukaryotic replication initiators are related with rep proteins. It's just the same. So it turns that the similarity between the plasmids and chromosome encoded replication initiators are somehow related with and more similar to specific similarities depending on the domains in eukaryotic systems you can identify. And that was just different concepts of this, how it happened. And of course, everything is of this last ancestor, and this could be convergent evolution, horizontal transfer, or non-orthologous gene displacement of AAA plus and wing helix fusion domains. So actually this is just tells you that the ancestor initiator primordial could contain primordial AAA plus domains, and Archaea would have AAA plus domains, and in gram negative bacteria it would be AAA plus domains and wing helix domains, and these wing helix domains would be fused in eukaryotic systems. Okay, then you can also have plasmid mediated lateral gene transfer from bacteria to this linkage, and that would just combining the archaeal and the bacterial wing helix with AAA plus to get the eukaryotic structured replication initiators, and also non-orthologous gene displacement by virus particles could be the reason, and the LUCA could have AAA plus and wing helix, and that would be transformed to eukaryotic cells and evolve to eukaryotic cells, and this displacement would just result with separate wing helix and AAA plus domains in bacteria. So different scenarios are possible, and you can just read about these different scenarios in here. Okay, so far I mentioned to you that in eukaryotic and plasmid replication initiators, there is two types of domains. Actually, one type of structure which is required for those proteins' interaction with DNA, and those proteins contain wing helix domains, motifs or parts which is important for the interaction with DNA. But to make the things more complex, as usually, in some replication initiators, which are listed in here, so also ORC or plasmid replication initiators encoded, or Homo sapiens, you see that on N-terminal part, you can identify from here to here specific regions which are usually intrinsically disordered, but they can bind DNA specifically, and they could be ordered due to their interaction with DNA. And this is very important. So intrinsically disordered structures are it turns that nowadays they are very important because they can just form the structure depending on the interaction with partners. And if they interact with DNA, they can form specific structure, but if they interact other partners, other proteins, then they just form a little bit different structures. So what we did, we just cloned this, we call this DNA binding domain, and using plasmon resonance, we constructed sumo variant of this, having wing helix domains and this N-terminal part, and we found that both wing helix containing recombinant protein and DBD containing protein interact DNA, and this is just SPR analysis which we mentioned before. And you see that there is increase of mass and increase of mass in here, but there is no increase of mass when we introduce specific point mutation in position 151. So this amino acid was changed. And also we did not observe in the control experiment interaction where the sumo alone was introduced on the sensor chip. So just make the long story, it was like 5 or maybe even more years project, but finally we published that the replication initiator contains N-terminal domain, which we call DNA binding domain, and this N-terminal domain present in here is also important for the interaction of a protein with DNA. Moreover, this domain together with wing helix domains, okay, wing helix, the green one, provides sequence specificity of the interaction. That was the long story. It was very difficult to find it because we just deal with intrinsically disordered protein. So we apply advanced bioinformatics, and also we constructed very many mutants with substitutions, and then we test the interactions, and then we just did the cross-linking studies with which we identified the peptides interacting specific parts of a replication origin of etheron sequence, which we modify to be able to cross-link those peptides, and then using mass spectrometry we identified this short peptides just to have a clue about the interaction of a protein with DNA. So based on this, we just figure out that there is a third domain, and those 3 domains are important for the interaction and site-specific, sequence-specific interactions of proteins with DNA. Okay, are we going to go further about the regulation? Afraid that not. I think that we will finish on the next lecture because that would be just the entire piece to discuss. So, okay, next lecture we will start with regulation of replication initiator, reinitiation by rep proteins, and formation of filaments on the DNA. Okay, so I think we can just finish in here. Thank you very much for your attention, and we are meeting next week, yes? Hello?
Next week is free.
It's free? Okay, so I'm just checking in the calendar. Maybe
Yeah, that's the
right.
Polish.
Yes, because it's 11th. Yes, am I right?
That's right.
Yes, it's free. It's okay. So within 2 weeks we will meet. 18th. Okay. So thank you for your attention. Do you have questions? Just to summarize, we discussed different techniques, potassium permanganate, the structure of replication initiation proteins, a little bit about the evolution. Yes, that was it. The structure, interactions, domains, this is evolution, convergent, horizontal, and non-orthologous, comparison between the different proteins. Then we also discussed the interaction of SNP, not interaction, but the specific terms. Okay. And the first part, potassium permanganate, and the role of DNA A and Rep proteins in origin opening. This is this experiment. And iterons and DNA boxes, so also this for the extrachromosomal genetic elements, and also the fact that the replication initiator has to be monomerized for the interaction with iteron sequence. Okay, so this is it. And the cooperative and non-cooperative interaction of DNA A protein or whatever protein, and how to identify this cooperativity using electromobility shift assay and EMSA. And this is just, I think. Okay, and the beginning of the regulation. So this complex story about the regulation. And so this is just a summary of the regulation which we discussed in the first part of the lecture. Okay, so thank you, and we are seeing each other not next week, but the other week. Thank you very much, and have a nice afternoon. Bye bye.
Thank you.
Thank
Goodbye.
you.